What if everything we thought we knew about cellular programming was backwards? A groundbreaking discovery from Cleveland researchers is turning the field of genetics on its head, revealing that two mysterious molecular switches work together in ways that completely surprised the scientific community. Here's where it gets controversial. When scientists tried to turn off what they believed were gene-activating switches, the genes actually went into overdrive instead of shutting down. This unexpected twist is reshaping our understanding of how cells decide their destiny, and it might hold the key to treating diseases that have puzzled doctors for decades. The research team at Case Western Reserve University has uncovered a fascinating partnership between two epigenetic markers. Think of them as molecular post-it notes that tell genes when to wake up or go to sleep. These markers, with the tongue-twisting names H3K79 methylation and H3K36 trimethylation, were previously misunderstood by the scientific community. No one understood how these markers work together, and now we have a mechanism, explained Dr. Kai-Shang Cao, the study's lead investigator and assistant professor of biochemistry and genetics at Case Western Reserve School of Medicine. But here's where it gets really interesting. Cao believes this discovery could lead to entirely new treatment approaches for both rare developmental disorders and common cancers. Published in the prestigious journal Science Advances, this research used cutting-edge genetic engineering techniques to systematically remove the enzymes responsible for creating these epigenetic markers. The scientists worked with stem cells from model organisms, carefully eliminating first one marker, then the other, and finally both together. And this is the part most people miss. Removing just one marker at a time barely made a ripple. The cells continued their normal development with only minor hiccups. However, when both molecular switches were eliminated simultaneously, cellular chaos ensued. Genes that should have been carefully regulated instead became hyperactive, completely blocking the cell's ability to transform into neurons. To our surprise, when we removed the markers, we found that the genes were overactivated, revealed Cheng Zhang, a Case Western Reserve graduate student who played a crucial role in this discovery and recently earned his PhD in biochemistry. This finding directly contradicted decades of scientific assumptions. Here's what makes this discovery so significant. Previous research examining these markers individually suggested they functioned as gene activators. Logic would dictate that removing them should silence genes, not amplify them. The reality proved to be exactly the opposite, a perfect example of how biological systems often defy our simplest explanations. But the implications extend far beyond academic curiosity. These epigenetic markers play starring roles in serious human diseases, particularly affecting the developing nervous system and various cancers. While their involvement in rare neurodevelopmental conditions was already established, their connection to cancer, especially blood cancers like leukemia, opens up entirely new therapeutic possibilities. In leukemia, the cancer essentially hijacks the body's blood-making factory. Defective methylation patterns can cause blood stem cells to rebel against their programming, refusing to mature into the specialized red and white blood cells our bodies desperately need. Instead, they remain stuck in an immature, rapidly dividing state that floods the bloodstream with useless abnormal cells. The research team identified a potential game-changer, a protein partnership called YAP-TEAD that becomes dangerously overactive when both methylation marks disappear. Think of it as a cellular accelerator pedal that gets stuck when the normal braking system fails. By targeting this runaway partnership, doctors might be able to restore normal cellular behavior in certain leukemia patients. What's particularly exciting is that drugs designed to block the YAP-TEAD pathway already exist and are currently being tested in clinical trials for other cancer types, including mesothelioma, aggressive cancer affecting the lung and abdominal linings. These medications were never previously considered for treating leukemia patients with methylation abnormalities, but this discovery suggests they should be. Dr. Cal emphasized that this treatment approach would specifically benefit patients whose leukemia involves dysregulation of both epigenetic marks. It's a precision medicine approach that recognizes not all leukemias are created equal. Different molecular signatures require different therapeutic strategies. Of course, the path from laboratory discovery to patient treatment involves numerous hurdles. The research team's hypothesis must first undergo rigorous testing in disease models that more closely mimic human conditions before advancing to actual human clinical trials. This process, while necessary for patient safety, can take years to complete. The collaborative effort involved multiple institutions and researchers, including Dr. Fulai Jin, a genetics professor at Case Western Reserve, who co-leads the Cancer Genomics program at the university's Comprehensive Cancer Center. Additional contributions came from research assistant Emily Cook and scientists at Fudan University in Shanghai, China. The U.S. National Institutes of Health provided funding support for this crucial research. Case Western Reserve University, where this breakthrough occurred, stands among America's premier research institutions. The Cleveland-based university serves nearly 12,300 students from 96 countries across more than 250 degree programs. Its strategic location in Cleveland, a thriving center for healthcare, business, and cultural activities, provides students with exceptional opportunities for hands-on learning, research participation, clinical experience, and entrepreneurial ventures. With over 125,000 alumni making global impacts, the university continues building on its legacy of innovation and discovery. But here's the question that should keep us all thinking. If we were so fundamentally wrong about how these essential cellular switches operate, what other biological assumptions might we need to reconsider? The history of science is filled with moments when conventional wisdom crumbled in the face of unexpected evidence, leading to revolutionary advances in human health. This discovery challenges us to approach biological research with fresh eyes and open minds. Sometimes the most important breakthroughs come not from confirming what we think we know, but from having the courage to question our basic assumptions when the evidence points in a different direction. What do you think? Should the scientific community be more willing to challenge established dogma, even when it means admitting we might have been looking at things backwards all along?